So good afternoon, everyone. And first, thank you, Jehome, for the invitation. It's my great pleasure to be here and to discuss some of my research. So my angle is from the other side, but we want to reach the same goal. So as Jehome just mentioned, so the title is saying everything, democratizing big data analytics. So we are entering a big data era, and probably already everybody knows. So I'm more from life science, biomedical science background. So we were deeply affected by that human genome project. So when the turn of the century, they sequenced the human genome, then quickly they followed by the microarray for understanding what's the transcriptome, then RNA-seq, then single cell, then epigenome, then multiomics. Then now what we really aiming to do is translate all this big omics data, the investment into cure disease or improve the crops or improve the environment. So we really are in the place we need to do the translation. And here is just what are the data we are facing almost every day, because I'm in life sciences, so almost everyone doing this omics data. 10 years ago, a few, now it's everyone. So there's no exception almost, unless you are hitting on the rock. So we are doing single omics, early days, genomics, transcriptome, proteomics, and metabolomics. This is where I came from. I'm doing some metabolomics and doing RNA-seq and microarray early days. Now every day is multiomics. So we talk about microbiome. Microbiome, you need to sequence 16S. You need to do a shotgun. You need to do metabolomics. You need to do proteomics. You need to understanding functions. And the other thing I'm doing these days is exposomics. Basically, we want to understand the environment. The gut is part of the internal environment. So how the genetics and environment interplay to affect your health. And we there's a lot of posts on spatial omics. So spatial omics also naturally combine a lot of things together. Food, how we eat, affecting our health. And on top of this, we have a lot of collection of phenotyping data. And in plants, they do a 3D imaging. They really see the leaves, the roots, and the growth, everything very in detail in 3D and high resolution imaging. And phenomics is mainly talking about clinical data and the patient history. We are talking about hundreds of the attribute for each patient, and the electronic record is for following their genomic sequencing, everything. So overall, we have tremendous data. How do we understand and making decision on top of that? So this is one really I'm trying to illustrate the issue. So we're talking a lot about we want to use leverage the big data, omics data, high resolution data to do the translational discovery. But the effect is that at least at the moment, it's so hard to do it. The thing is that it's not just a few elite group who have the huge team of expertise who can analyze the data. The translation actually in the majority of researchers every day, small lab or medium lab, they really need to testing, do the hypothesis-driven test research, and do the validation and push it to the like say clinical translation and stuff. So if we really want to do the translation, we need to address the big data challenge, bring the data accessible to every researcher. So, and if we can include more ordinary people, that's better. But at least even within a research scientific community, we have only an elite group, like 5 or 10% of the researchers. Majority have no expertise. So how do we actually deal with this? I feel very how to say it's obligation for bioinformaticians, about people who's doing bioinformatics, need to really addressing this issue, just because bioinformatics naturally interfacing between biology and computer science, and dealing with data and making data more accessible. This really that's the task. It's a whole field as coming to the first place. So so how do we do that? And I'm being in this field for the last, like say, almost 12, 15 years. So how do we actually deal with this? And we don't talk about the general solution, but the solution is actually abundant, and almost we can do it very easily. There's a lot of patterns that we can borrow, actually solve a lot of our daily struggle. And some of the things I put it in a high level. One is appreciate big data, and big data actually a huge benefit, especially for people who have computational skills and a statistical background. And we just need to appreciate embracing it, and also need to, when you deal with big data, aware of the context. Some biology background, how it's going to be used is critical. And also keep in mind the end is not a publication, the end is about translation. So a lot of times you need to really think about uh, the goal. So once you have that cycle, actually it's become very how to say, um, the if you're talking about a game player, the rewarding loop is very strong. You have the tools and being used users who making discoveries, and really it's very good loop. And how do we address the big data? Is big data come with a lot of the progress at the simultaneous, almost same time, the cloud computing and a lot of the hardware improvement. And about 10 years ago, we don't talk about just a few gigabytes of RAM. Now we talk about terabytes of RAM, and cloud computing everywhere. So most time, the, the hardware is not limiting factor, is our ideas. So and a few of my slides is actually talking about this, how do we really understand mission of bioinformatics, is that at the end, at the first place, it's aimed to address this big data production. We have this high throughput instrument, like next generation sequencing and mass spec. We generate tremendous data, and this data need to be really processed properly. And we need to have database, we need to have tools, we need to have algorithms. So in the end, is we need to understand data, making translation. So all this in between, we're talking about this is bioinformatics. So bioinformatics need to really keep in mind this is the mission. And how do we actually bioinformatics, who are the bioinformaticians doing the everyday job? And I see a lot of people, maybe mostly you from computer science, and I guess, and a lot of people also from the other side, the statistics, and a lot of people from just medical life sciences. They just start analyzing data and feels more rewarding, and starting developing tools, algorithms. So overall, the bioinformatics have a very interdisciplinary nature, and it's very fuzzy. So people working with each other need to be aware of this different background. Overall, solving a problem need to really understanding each other. So bioinformatics, naturally, you cannot treat them just purely like computer science. Then that's going to be a hard to address. The other part is bioinformatics really need to understand the users, and who is going to receive the result, who is going to use that to making decisions. And a lot of the big data, and you cannot directly send to your collaborators or your end users. You need to process them. And a lot of times, process and need to help with statistics, machine learning, and now we talk about ChatGPT or AI or a lot of other things, and visualization is very critical. And the end user need to, usually they have the expert in certain like cancer immunology or certain things, they have the domain knowledge. But a lot of times our brain really cannot remember everything. So we need to assist with domain knowledge. So we need to have database, have the AI, have a lot of the things to support that. So the background, context, and the data need to be processed, and we need to have the stats, we need to have visualization. So the user in the center will need to be informed. They need to spend time, start thinking about this. So overall, it's really, we're talking about a packaged the deal if we think about how we deal with the big data. And also, if we focus on the big data in life sciences, we're really talking about just various omics data. Actually, omics data is very, how to say, became very everyday, and a lot of things, the main backbone is really routine. So in my case, and I'm dealing with mainly two type of data. One is sequencing data, next generation sequencing. The other one is mass spec. So mass spec, big data, next generation sequencing, big data. They are very different. You need to process them differently. I know there's another one is imaging data, which so far I haven't touched it, but overall that the data actually can be categorized into really main big categories, and you can process them using some very well-established tools. And so that's the data processing. After that, we get a matrix. Now we can apply a lot of machine learning stats, and we do the comparison class. Learning is more or less starting from here. So this is all the computer science stats should be countable. And last part is slightly more to biological. So pathway analysis, gene ontology analysis, and enrichment and network analysis. So this is a slightly more biological, but the first one is more machine platform specific. The second one is really data science. Last one is more like biology or function. So this is just almost shared across all the omics. And that's one thing. The other part is that we need to understand that when we deal with the raw data, it's very big. So, and it's usually you cannot deal with in the real time. We need to have several hours if you talk about large data. Even it's high, you need to submit and wait and get there. But when we do at least a general stats machine learning, even it's a relatively big matrix, we can get it relatively fast. But the issue is that you cannot give user the answer. This is the things. It's because it's very complex, and each user have different goals, and we cannot give them just a numerical ranking or just this is the classification accuracy. And you need to deliver the result in a more friendly way. It is very complex. You need to understand a lot of features, false positives, and interpretation context specific. So the thing with here is that omics data need to be interpreted within metadata. Clinical metadata is very important. And also data conform with the published result. What has a general understanding of this pathways stuff. All of this is public knowledge. So you need to double check the result, making sense or not. Program don't need to do the checking, but you need to provide it to users who will make the judgment. So this part is critical for solving a big data problem. So the other part is that omics data is different from like a well-designed target-based data. It's mainly exploratory, and a lot of purpose is hypothesis generation. So in that case, you need to give a lot of freedom to the researchers who can actually spend the time exploring and try different ways to understand it to their own kind of answer their questions. So the requirement for this is very different from like individual staff or system biology. You need to do really strict check and just run the whole thing. But omics analysis usually is open-ended, and what the main thing is patterns and trends, and probably more prioritize certain biomarkers, and for people to think to making their decisions. So I mentioned about the big data, and how do we deal with this is be aware and be friendly. One thing is computing friendly, and my lab, we deal with a lot of data, and for the raw data we usually we use Computer Canada, but we also have our local server. So we just mainly doing it locally. We submit to do the job, scheduler, they will do it just 24/7. And we don't usually use cloud. Data to the cloud takes time. So that's the thing. But we use the cloud to do a lot of machine learning computing. And because once you have matrix, it's much easier. And the cloud actually very, very robust. We never have any just failure because of cloud. But we do have several issues once in a while. So that's what I'm saying. Cloud is a really, really to-go choice. The other one is biology, and we have a lot of data, especially omics data. We need to keep in mind is that in our normal human or a lot of physiology conditions, we have homeostasis. So we don't expect a lot of things to change. We only expect a small portion to change. There's a lot of hypothesis testing based on. So in that case, a lot of the data can be filtered out without losing any important information. Actually, it improves the signal and improve the power. So based on this thinking, some people suggest almost half of the data can be filtered out safely. And you leave the data that's more informative. Okay, so a lot of times is how do you process the data at the beginning, and the result will be more robust, more informative. And this is when it coming to the what is result look like. So result is really delivered to the researchers who actually facing it, want to do the understand it. So you cannot give them an Excel spreadsheet, you open it and see all the numbers. Our brain is really not good at understand numbers. Even computer feels clearly, we don't. So we are good at patterns, we are good at coloring. So one of the things I found it very useful is like heat map. I think a lot of the uh, previous slides already show in the other presenters. So here's a heat map we used quite frequent. So you get all the values converted to a heat color, and you can do the clustering. And now use mouse to zoom in and just drag and select. And this select the region is becoming your point of interest. Why you select that? Because you see these are four conditions and they have the regions that are very different. You're naturally curious about that. You select the region and you do an analysis of what's the functions about this. So it's very natural. You just do it and you feel satisfied. So this is part of, if you love biology, this is part you feel rewarded because you see the pattern and you understand and you see the pathways and you feel it. So this one is very simple. And I'm just from why we do that is because we want to do that. And from biological point of view, it seems very natural. And here's another part we do is use networks. Networks can do higher level summary. So if you're doing a heat map, you actually see individual values. Once you see the pathway, you can actually go to higher level and how the each node works. Each node can be a sample, can be a pathway, can be more high level things. And you can see actually interactions and based on the connections or other correlations. And you see layers of layers interaction. And again, you can actually see what's patterns and select it and find more. So we call it called visual analytics. I know this is a lot of people think about different ways, but I found it's very intuitive. You see it visually, you do the analysis on top of it, see it, analyze it, and direct it there. So it's very encouraging people to explore their data, especially for this omics data, which we don't have a how to say a very clear idea what exactly we're looking for. So we want it to be more exploratory. So this part is we're talking about how the general framework. We have raw data locally. We have a matrix on the cloud, a lot of data visualizations on the browser. And finally, we have the researchers who actually interact with the data. So it's big data and complex data. We try to address that. So this is tools. We actually have almost 20 different tools developed. And this is just a few of them we put here. We have this is probably more this year. Last year, we have almost close to 1 million users using our tools, and it's continue growing, and it's clearly scalable. And every day we have about 5,000 users. 5,000 users. So during regular day, we have about 200 to 500 users simultaneously. So because we know that's um, how our in the back of our server. So I'm just described our general approach and our tools. Now we just show some case studies or how in particular case studies, how we deal with that. And I just removed the metabolomics, which actually almost half of my research focused on that. But I guess this audience is not usually doing that. One I'm going to show is next generation sequencing analysis application to non-model species. It's a very unique challenge. And then we're going to discuss about multi-omics. Multi-omics is really cutting, not to say cutting edge, it's really a hot topic. People are collecting a lot of data, how they going to address that. So this one is, I call it NGS omics because it directly deal with next generation sequencing short reads. And we deal with that in human and mice. And if you sequence to the send to the genome Quebec and they're going to sequence it, they're going to mapping it, or you get it almost a nicely formatted tables of genes and their abundance. You can do a statistical analysis a lot. It's very convenient. But if you study non-model species, say about frogs, birds, and certain fish, and it's so hard. You sequence them, you get all this, spend like 200, not 200, $20,000 and sequence them, and you get terabytes of data. So what? You have sequence, and you don't have biology. How are you going to get biology? You need to do sequence assembly. And in order to assemble, you probably need to combine the short reads, get long reads, and try to do sequence assembly, and which takes tremendous computing power. And you need to use collaborating with somebody who's expert in doing that. And it's running almost several months, and almost in my experience, it takes probably a year to get some assembled decent, and you need to annotate the assembled genome, and if you do an annotation, you're going to find out most of them is called a hypothetical protein. So it's still not informative. You spend so much money, spend so much time, and still you don't get in decent biology. So this is really, really frustrating for people who study non-model species. Even we get their all the sequence reads, we get their sequence ATGC byte by byte, but still we don't get biology. So what we can do, and at the beginning we try to do assembly, and we realize it's not scalable, it's not going to work well, and then we have an idea. So we have the short read is actually not so short, 115 base pair or 100 base pair. It should be actually quite sufficient. If you really translate it into like 13.3 or 13.6 amino acid, and you map into the back, and uniqueness is almost 99.99. Yes, there's some repeated regions, but majority will be fine. And we are studying our model species. We are not looking for that corner cases. So what we think is we can directly map the short reads to all the genome across all species. Okay, it's just we think it should be doable. So we started doing that, and yes, then we actually using or just translate search. We just translate 100 base pair back to their amino acid, and using 6 reading frame and directly search an Autolog database by translate all the transcribed translate how to say all the coding sequences and to the as a database. So once we do that, actually the computing time and the performance is very good. So we can get almost very good speed and memory control, and from weeks and months we can go to actual hours, and we continue to improve it. And now we actually can directly search directly at the not don't need to translate, especially now we're doing microbiome data. Sometimes we already have the hit in the database. We don't need to translate. We directly mapping back. But if it's no map, we do the second tier of translated search. Overall, the speed can be faster. And so we almost have a unified, unified RNA-seq analysis for microbiome also. So overall, is that we can do almost data analysis across all the domain of life, and we're able to actually mapping them across different species, and do functional analysis. So this is quite something we're proud of. So it's just been going to be on Nature Communication probably soon. So overall, that we didn't really just do an algorithm. We put it on online and make sure people can use it. And also you can download that Docker to do that. So we also use a tool called Callisto. If you really have that reference genome you want to use, and you can use it. But if you don't, and it's also doing fine, the same speed, and the resolution actually quite fine. Now I'm going to talk about multiomics, and I think last one is talking about non-model species next generation sequencing. Now we're talking about multiomics. We have so many different data we want to integrate together. How do we do it? And if you know solution, we just manually do it. Why? We just analyze individual data, and we think in our mind, and we still can write a good story and a good publication. But that's really not scalable, or it's not objective, and people have different background or get different interpretation. And what's the better approach? One is called knowledge-driven. So knowledge-driven, I'm going to discuss next. Then there's data-driven, probably more objectives. Directly, you have the data matrix, how you combine them. And there's a lot of discussion this morning talking about dimensionality reduction. I think this is one of the things we are trying to conquer, make it more accessible, because it's really complex. And in the future, I hope will be AI driven. You just talk to them and they'll be interactive with how back and forth until data start making sense. So knowledge driven, knowledge driven integration, what it is, and it's actually very simple. We just call it connecting the dots. And individual data, you doing a normal analysis, you're going to find the signaling features or patterns or biomarkers. This is called the seeds. You need seeds, and you can actually doing this for each omics data. Then we try connecting them. How do we connect them? It's connecting using our background knowledge. So if we have this background knowledge in the computer, and we can do almost graph mining, and within the graph, that's integrating all different seeds and presenting them in a coherent way, and you will see all these genes and metabolites or proteins actually participate in the same pathways. Then that will be very intuitive and explainable, right? So this is exactly what we're doing. So it's idea simple, but do it well and let everybody can use. It's not so easy. And so this part is we just talking about this is idea, and we want to do that from SNPs, from genes, from proteins, from metabolites, actually also from the peaks. This is almost raw data level. And why we do this for peaks is because I'm doing a lot of time on metabolomics, MS, which is far behind the RNA-seq. So I spend a lot of time actually on how to get raw data accessible. MicroRNA, transcriptional factors, microbes, this is microbiome data. So we have them, we need to translate them to be more connectable. So then we have a database. You can think about this as a graph database, all the things connected. Then you try to map them. So the idea is that it's not just the mapping and you talking. We really want that result map to be intuitive, to be interpretable, disregarding who's using it. We cannot just generate a pretty image and put on publication and people say it's so good, but how can I do it? So our idea is not we do it, it's just you do it. And we have these tools, and you copy and paste your list of genes, proteins, and metabolites, you submit, then they try to connect. After connect, you can customize, you can visualize. And you see we spend a lot of time doing the 2D, 3D. Why we're doing the 3D is because at least a few years ago, we have a strong belief there's going to be VR resolution. We also bought that Oculus Rift. We really can put it in a virtual world. We can have this touchable stuff. It's doable. It's just we found out there's a lot of things to continue to improve, make it more cheaper, probably free for students. And we can do data analysis in a virtual world that we can collaborate in. So making a network of information on the 3D is technology is doable, it's hardware. And so this is all cloud, not cloud native and web native. So we don't need to install any plugin. It's all native for the modern browsers. So here is that you can upload your data, and it will create a network, and you can visualize it. And even it's complex, you can actually perform module detection. Module detection is basically collapse things that are similar to together and have a bubble and label together. And when you click on it, it will come to you, and all the background going to fade out. And you can actually doing more analysis on top. So this is really get the focus, and even there's big background. So we can also do a 2D, we can do a multi-layer. So this is all kind of natural. And next one, I'm going to be on this model-driven. And the model-driven is harder. We have to try to attacking it multi times. Why is that? Knowledge-driven is more about visualization, and it's intuitive, and people don't need to think too much. It's just to see it, you kind of understand it. But model-driven is really hard. It's math, really dense. We talk about all this derivatives, all this calculus, all this matrix manipulations, all stuff, and it's probably intuitive to most of you, but it's scary for most of the researchers who really want to just use the data. You don't want to see any formulas. So that's really how you tell them this is a pattern, and they want to see the what's evidence? What's behind this patterns? So this morning, they talking about explainable AI. So first one is talking about the gradient, which is making sense, but it seems hard to get a consistent calculation of the gradient, which is because it depending on location. The other one is talking about visualization, which is exactly what we are doing. We want to just give as much as information as possible if you desire so. So visualization is critical to be transparent, to show the evidence. Some people have different perceptions about it. I understand this, and probably not meaningful to you, but I understand that. So more or less back and forth, you learn because you see the pattern. And this part is called omics analyst. This is initially we designed it as a 3D room. People get in and put their data in the middle, and you see this side is network, that side is the heat map. So actually, it's doable, and we can try to talk to Borderland and make it in a more science. But the important thing is really is how to make it people can understand, because this is not a game. This is actually data and presented as it is, but make it more, how to say, distilled in a form that seems the pattern is clear. We continue to upgrade it. We just want to make sure it's as simple as possible, but it still make it flexible, but still you can use it easily. And how do we do that is first data upload. Multi-omics data need multiple matrix. So you need to upload that matrix. You need to upload the metadata, describe what's the patient, their age, gender, medical history, all this stuff. Then it's like genomics, transcriptomics, metabolomics, a lot of things. And so far we're supporting about 5 different data types. We don't support more. Why? Is because we don't really see that more than that number. So after that, we do some regular data analysis. There's a lot of clustering, machine learning, and dimensionality reduction. And after that, we use networks and scatter plot in 3D or 2D is all fine, and networks to really show the result. And here, basically, this is some of the things we show, but there's a lot of improvement in the last few months. So this is one is we show the patterns of separation, and to see it in a 3D, we actually can directly make it more abstract, and then we see each groups where they are and the features who's contribute to the separation there. So in the PCA, this is called a score plot, this is called loading plot, but we can put it overlay on top of each other. Then you get one totally, almost you have, don't even display or just have one pixel, you don't see it, but you still see the big patterns overall. Then on top of that, you put these features there. And here is a correlation network, and you can overlay different things. Overall, our goal is to make this dimensionality reduction, deep learning result to be more accessible, and people can trace back from the patterns back to layers of evidence earlier step. So at some point, they will feel satisfied. They will feel they understand the data. Even sometimes you don't know you really understand, but after several times, they will feel they understand. Eventually, they will be comfortable using the tools. As long as being transparent, reproducible. We are scientists. We believe that's logical things. We give up. Maybe we just use it continuously. So overall, as a multi-omics, we're talking about is more complex, and what is the best thing we can deliver the result and make people understand and making decisions. And I think there's no magic solution, but we need to engage in the users, really understanding what making sense to them. The other ones, we need to provide more contextual knowledge. Context knowledge, basically, the certain pathways, certain like annotations in the background. Once they click, they want to understand more, then they give them more. The other ones deliver them to them in a transparent way. So also fast. If I'm in a mode, I want to understand the data, and you tell me 3 months or 6 months, I probably already forget. So I want it, and probably I get it now or get it tonight. So I continue my thinking, my idea is still there. So I'm doing it. So the one of big data is need to be delivered fast, and you get the features, you get biologies. So you continue to improve the circle, back and forth, back and forth. So once in that circle and back and forth, a few rounds, and people will get it. So I think that's the goal. We don't expect a magic solution, but we expect a collaborative approach. And from like a tool development point of view or bioinformatics point of view, what we need to do is educate. And so we need to try our best to reach the other end, but the other end also need to improve their knowledge, like basic stats, basic design, or basic machine learning. So meet in the middle, right? The other ones engage and empower and enable. This is all about how we make data and tool more accessible. And there's no boundary, because we are doing the same research. Eventually we want to translate. So they are the one actually doing the translation, and we are doing probably make things accessible so they can make a decision to the next. So more or less we are doing together. And if we have very clear boundary, we are doing data science. After that, I push that data to you and don't touch it. But overall, the whole cycle of data processing and eventually translation and the drug discovery is very satisfying. And this is the last bit, and why I'm trying to talk about citizen science is we feel it's not tool development, the technology is already, but it's the community, it's education. It's really how can we harness the power of the community? And ChatGPT really surprised everybody. I think I'm so surprised by the performance and how it does. But before that, I actually really actively tried developing a chatbot supporting omics data analysis, because our tools being used by so many people most of our time. It's not actually developing tools to actually answer their emails, answer their questions. So we put our forum, and so users' question can be answered not by us or also by the other people. So the answer will feed into other people who have similar questions. So it helps. And but we do feel the ChatGPT like things can help a lot. So overall, that we talk about what's the next generation. It's called conversational analytics. Conversational analytics is what you need, what's the data about, what's your questions, and back and forth, and people will really get most of the data. And it is actually doable. And almost 2018 or 2017, we tried a chatbot. It's more or less doable. It's just frustrating at the beginning to really communicate your needs to that chatbot. But since ChatGPT really solved that well, but we don't know how we able to use ChatGPT for this situation. We are still learning. So what's the future? I've just put this one. This is I think it fit well for the talk. It's a more AI-assisted self-service omics data analysis, and really you think about what ChatGPT can do, and with the regular, just a conversation, very loosely connected things, they can do it accurately. Omics science is actually, it's hard, but it's so structured well. A little bit of training, we can get it almost so accurate. So once we get that first layer, understand the user needs, linking back to the different algorithm visualization, it's almost so straightforward. That's at least in my point of view, is it's always the front end that get the user needs. The back end, it's more or less it's done very well. So it's that's the thing. This is my last slides, and my team. Thank you.
Thanks, Dr. Next question.
Come on, questions. Yes.
Any interesting examples of how your users have used these tools to reveal insights that maybe otherwise wouldn't have in data?
Yeah, I guess a lot. It's
But anything off the top of your head?
It's a good question. I'm being asked actually to come up summarize who's that. I do see a lot, but yeah, it's still probably early stage. But if you see the people who's using our data, publishing, using our tools, publishing high-ranked journals, it's a lot, hundreds or thousands of them. But it's yeah, in terms of really getting to the stuff, probably waiting a few years, I think. But whether they are talking about early, leading by these tools. I really doubt it, right? Yeah, but it's a good question. But I do interacting with a lot of researchers who are using our tools to making discoveries. That already is very satisfied because they are clinicians, they are frustrated, and they have certain needs and tell me how the tools help them get what they want. That's very good. Yeah.
So I know you mentioned that for the NGS data, the kind of initial primary analysis is done locally because data upload for that much data is challenging to the cloud. Do you foresee that kind of moving in the direction of all being on cloud at some point so that users can potentially reanalyze from the initial step or
Yeah.
kind of do their own aggregate analysis with their data from one step and that sort of thing?
Yeah. The framework was done was about 8 years ago when the internet speed is still low, and nowadays actually it's not that big deal. Everything can be done on the cloud. I'm just letting you know that's so true. And I feel you can use cloud for almost everything. The thing is that we use it local is because we have the server, so it's more or less free. If we're using cloud, who's paying the bill, right? That's the thing. So we didn't open that because if we do that, we need to let the user to really link to their credit card. It's still taking the computing cost is not negligible. We are talking about thousands of users every day, right? So that's the issue. But yes, the bandwidth is improved a lot in the last few years.
I may have questioned myself more. So you're developing all infrastructure. I would like to hear a bit more about the all efforts you may do in terms of training or new people coming, how they can acquire the expertise better using the tools. You spoke a bit about the forums and the ChatGPT also there.
Yeah.
But are you also developing other activity tutorial training?
Yeah.
And how much effort it requires from you? So I would like to see basically if you put that the time you spend about this kind of training and the one about building the platform in general, how you will
almost half. Nowadays, probably changing slightly more even. We publish a lot of tutorials. We just submit Nature Vertical on multi-omics because we really wanted it accessible. So it's very detailed stuff. But we also host a workshop almost twice a year. So probably we're going to get it more often. And the thing that people want more hands-on. And so training workshop supporting it became quite important. It's not about too much about tool developing. Once you're developing there, you need to really let people use it properly. So at the beginning, tool there you have some bugs, but soon it's not about bugs. It's about how to let user know, understand the data, understand tools. We do writing like tutorials, but the best time actually have a workshop, and they will attend, and you can see them, you can interact with them. So in addition to forum, so it is it's a whole package almost. Yeah.
And are you developing tools to efficiently imagine that sometimes some data sets that there are being processed could be also relevant for research for this? That's basically some mechanism can put in place to promote collaborations or exchanging data or expertise analysis. Are this mechanism in place?
Yeah, this is a suggestion I received from people with similar background like you, even a few years ago, and I really found it tempting. I really want to do it, but on the other hand, it feels we don't know how. When you save so far that we don't save user data, you come and use it freely, and we're going to remove just transiently. It's very clean, but if you want to keep the data and allow you to collaborate and share the data, the first is the privacy things and how you maintain it, because every upgrade. So there's a lot of the thinking on that, and it's definitely something in my mind. I think we can leverage that. There's two things. One is data. One is actually how they do data analysis, the workflow they choose, because our tool is actually quite comprehensive. And for this data, what's the best analysis? There's a whole workflow. We can capture that. So we can capture the knowledge, how they do the data analysis. We can also get the data. And we just not sure we are ready to harness that because we need user agreement, right? And we need to really, we know we can make it into good use. So without violating some privacy concerns. So it's very good, a good one. And I think, yeah, if I get good ideas, I will proceed. So far, that's still debating. Yeah.